Welcome to the Rich Report, a podcast with news and information on the world of big data. Today, my guest is from Bina. We have Narges Bani Asadi, who's the CEO and founder of the company. So, welcome to the show.
Thank you very much for having me.
Oh, it's my pleasure. Now, I brought your slides up, so why don't we start with that, and then we'll follow it with Q&A.
That sounds great. So I'm Narges Bani Asadi, the founder and CEO of Bina Technologies. We are an early-stage startup based in Silicon Valley, California, and we are very excited to be part of this podcast. And actually, we are just launching our company and have the very first public review of our technology this week, happening at BioIT World Conference and Expo at Boston. And we are talking about our very first product, that is Bina Box. And we'll tell you more about it and what does it exactly doing later in this podcast. So our mission at Bina is simple. We are here to solve the data analysis bottleneck in genomics. And we are creating the fastest data and compute platform for analysis of genomics data. So why this is important for your audience, why this is a big data problem, and how it is related to high-performance computing. Genomic data is an extremely interesting data, very informative about our health and disease. And as we are going to see in the next few slides, it has become very accessible in the previous years, and now the challenge is how to analyze that. So before I go to details of what we do and why it is very important, let me tell you about how Bina got started. So I was a PhD student at Stanford University and studying electrical engineering and high-performance computing when I realized about the interdisciplinary projects that were happening between several departments at Stanford, including medical school, statistics department, and computer science, and the goal was to understand cancer at a very molecular level to see what goes wrong when the protein and other small molecules in the cell start some miscommunication, and how it can lead to malfunctions of cells and eventual onset of disease, including cancer. So we developed a lot of statistical methods and algorithms, but we realized that it is very data intensive, very compute intensive. It would take us weeks to run them on big Stanford supercomputer and clusters, and we realized it is basically a computation problem. And my task became how I can accelerate and make this computation faster and more precise at the same time. So this is how we realized that three words are kind of collapsing and correlating. So if you go to slide 3, we are going to return to slide 2 later. So if you go to slide 3, you will see how three words are kind of intercorrelating in solving this big cancer research problem. We have statisticians trying to understand the data. We have medical researchers who are producing the data and also take care of the very last stages of interpretation of the data, and we need computation to make all of this research happen. So you have high-performance computing, statistics, and life sciences, people all trying to work on the same problem. And what we realized is that these people usually do not speak the same language. Over time, people have become very expert and they have deep knowledge of one discipline, but it is very hard for them to think outside the box and see it from the eyes of the other disciplines. And sometimes the same term have different meanings. And for example, the life scientists, the people who know genomics and algorithms, they don't like necessarily to write good software. Or people who write very good software code, they don't like to know about the hardware and all the complexities of, for example, GPUs or FPGAs. They like to have these abstraction layers. And while these abstraction layers are very good for flexibility, and sometimes also kind of freedom, but it is not very good when you want to have a product that solves the end-to-end solution in the most efficient way. So what happened in the course of few years at Stanford was that we developed, I think, a new language so that all of us involved in that research project be able to work with each other, and I call it Bina language. And myself, I personally bridged between all these three disciplines. I had to learn a lot about statistics, biostatistics, and genomics eventually. And we thought it is a very important expertise to be able to bring together these disciplines to solve very important, meaningful problems of our era, that is healthcare. So if you go back to slide 2, I would talk about what is our vision at Bina. We think that healthcare is becoming transformed. So we are going to move from the inefficient, ineffective medicine of today to a personalized preventive healthcare where targeted treatments and prevention will help individuals to have much more knowledge of how they can treat diseases. And in this transformation, computation is playing a very central and important role. We need to look at many sources of data. Our lifestyle, our diet, our clinical history, our family history, and genomics are playing a very fundamental role here. It is basically the blueprint of our cells and guide how we are going to respond to different situations. And analyzing that data and know what it exactly means is very important in that transformation. And that is how we want to play a role. So Bina Technologies is founded to bring to table the expertise on high-performance computing and big data analysis to the world of genomics and really help cancer researchers and eventually clinicians to transform the medicine of today. So let's go to slide 4 and look at what is happening, why this is the right time to think about this problem, why people didn't start kind of this work earlier. The main reason is that for the very first time in the history of mankind, we are capable of digitizing humans, meaning we can measure these 6 billion characters on each individual genome in a precision and in a throughput that is unparalleled before. So this graph, what is it showing? It shows how the cost of sequencing or measuring the DNA sequence data has been progressing or dropping rapidly over the last decade. And this graph is in logarithm scale so that you can fit it in one graph easily. So if you look at this, you see in September 2011, it would take us $100 million or more to sequence one DNA. And it took us several years to do that. As of now, in 2012, we are capable of sequencing the whole human DNA with less than $1,000 in shorter than 1 day time. So this is millions-fold of progress that has happened only in a decade. And this is only the beginning. So this type of data is making us capable to study and understand human disease and health in a very granular, detailed level, and that will completely transform our understanding of how diseases are going to be developed. So this opportunity and this very valuable data brings by itself a lot of challenges as well. And all the challenges are about how to analyze the data, how to interpret the data. What does it mean to have these mutations or that variations, for example, in the genome? How can I share the data? How can I have the privacy in place so that people feel comfortable by putting their data out there? And that is kind of the new focus of the whole field of genomics is around big data now. And to just give you an example of how big is big data, we just hired a couple of engineers from Google, and they were telling me that the problems they see with one single genome, the kind of challenges they see involved in analyzing one genome are equivalent when you have an iPhone app or a Google app of having thousands of users at least. Because if you just look at one genome, it can easily be 300 gigabytes of information. And if you want to put it in the context of a population of patients, if you want to include other sources of data with it and look at everything, the big picture, it is a very data-intensive as well as compute-intensive application. So we think the world of high-performance computing can bring a lot of value to this field and help this transformation, and it's a very impactful and meaningful application of big data analysis. So let's go and move forward to slide 6. So I would like to speak about the different aspects of this large data. As we were just saying, the data sets are very big. Data flow is a critical bottleneck, whether it is transferring the data from lab to the cloud, whether it is from the shared storage to the compute cluster, whether it is the storage, the disk, or even the memory. And the type of software system we are developing here is trying to address all the data flow bottlenecks. And at the same time, it's very CPU intensive. There are trillions of string comparison happening all the time. It's all about these short strings of DNA that you need to put them together, kind of solving this puzzle, and then find the relevant mutations that's happening. So it's very compute intensive at the same time. The current solution, how people are trying to tackle this problem, are very inefficient. It's all based on academic open source solutions that are, some of them are very accurate, and most of them are very flexible and transparent, but extremely poor implementation in terms of scalability and performance, and not much kind of production level code out there. And the other thing we are realizing is that this is a big data problem that is extremely parallelizable, and which is very good. We can take advantage of, for example, distributed systems or cloud solutions. But at the same time, we looked at Hadoop or SunGrid engine solutions, and we think that these traditional approaches that have been optimized for web applications fail in addressing the unique challenges of genomic data. So I would like to introduce you my team. Three of my co-founders, Professor Wing Wang from Statistics Department at Stanford, Professor Gary Nolan from Medical School, and Professor John Warsnick from UC Berkeley. And actually 4 of us got together in 2010 and really decided to transfer our methodology that we have developed at Stanford to Bina. And you see the rest of our team here. We have a lot of expertise in different fields in our development team, as you see, from statistics and computational genomics to high-performance computing, distributed software, as well as UI and design, which is necessary if you want to make this technology useful to the clinicians and biologists at the end. So if you move forward to slide 8, slide 9, slide 9 is about our product. So what is our product? Our product has 2 pieces to it. There is an appliance, and then there is a cloud. So if you look at the top of this image, you see the series of steps that need to happen in order we go from a sample that is prepared in a clinical setting or in a lab, all the way to the interpretation and actionable items from that data. So the very first 2 steps happen in the lab. There's the preparation and the sequencing experiment. And after that, it's all about data analysis. The very first step, there are very first few steps that we call them as a group variant analysis. And that means how we find these variations in one person's genome that are unique to that genome. For example, comparing to a reference genome that we have, all of us share more than 99.9% of our genome. And all the things that go wrong are in that few million points in the genome that the mutation, insertion, deletion, structural variations, copy number variations, and all these things can happen. So that step includes like 10 different sub-steps, and it's very compute intensive. To give you an example, we are working with a big research institute, and they have about 1,000 CPUs in their cluster, and it takes them more than a day. They have parallelized around 1,000 CPUs, and still it takes them more than a day to run this step for only a single genome. And what we have achieved with our appliance, we have brought down that time to only a couple of hours using only one appliance, that is a 4U server. And then the next steps are about the interpretation, annotation of the data, and eventually comparative genomics when you put in the context of a bigger pool of genomes. So what is unique about our approach? There are two things that are unique. The first thing is that we are taking a very holistic approach to computation optimization. We are optimizing the algorithms, software, as well as the underlying hardware and implementation. The other piece that is very unique to our approach is this hybrid approach to cloud computing. We think you need this Bina box that is a cloud gateway to sit between the raw sequencing data and the cloud, because the raw sequencing data is extremely large. It is terabytes of data. It's very impractical sending it to cloud services given the current and network bandwidth available in hospitals and clinics. So with buying our appliance, they have this cloud gateway that helps us reach much bigger services through this box. So if you go all the way to slide 12, we have, I would just share this as one example of the results we have on the pipeline. So this is kind of how much it takes, how much time it takes to run this very first step that we were talking about, variant analysis on Amazon Cloud. And we see the different, like the sequence of steps that you need to take to do variant calling for a whole human genome data. And it takes about a week to finish that on Amazon. And this has been parallelized on one node, 8-core node, a high memory instance of Amazon. The last two bars that you see, Bina Pipeline and Bina Box, is our solution. So our software solution on Amazon is about twice or more faster than other standard solutions. But if you run it on our appliance, it will be extremely faster. So you can finish the whole analysis in only 2 hours. So how we achieve this speed up, again, as I said, it's a very holistic approach, optimizing many layers and the system software as well. So with that, I would like to end. I think I went over a few minutes of my time as well. What I would like to end with is that we are just announcing our pilot program today, and we are receiving applications from a lot of clinics, diagnostic labs, pharma, and research centers that would like to test our box and see if they like and they become hopefully customers very soon. So by that I end my talk. Thanks so much for having me. I would very like very much to receive questions.
Well, great. Well, thanks for that, Narges. This is very interesting. I'm curious about your market. You said that like clinics would be the pilots. Have you sized this market? I mean, how many clinics out there would be interested in this kind of product?
So, we are at the kind of very interesting time in this market. It is an emerging market, so it might not be very big today, but we are betting on the fact that it will be extremely big in the next couple of years. And what is happening is that because you saw this graph on how drastically the cost of DNA sequencing is dropping, that actually makes everyone capable of sequencing their genome very soon. And the patients and the consumers will drive this because of cancer. Because if you know what mutations you have in your cancer tumor, it is already very informative for which kind of treatment you need to pick. So as we're speaking, there are only about few thousand genomes out there, but the prediction is that by 2014 there will be at least 10 million genomes available for study, and the market is just exponential growing in the next few years. And to talk specifically about what is our market, as I said, we see these different segments. We see most of it is still in research institute and government labs who are starting very large initiatives on genome sequencing. And after that, there are several diagnostic labs with focus on cancer and some rare diseases that are really caring about turnaround time. What I will always say is that in our industry, saving time is saving lives. If you think how fast a cancer tumor grows over time, if you can cut any single day from that process, it is very meaningful for anyone involved in the process. And we are seeing already that several diagnostic companies and pharma are interested to become our pilots.
So walk me through this, Narges. If someone had this kind of capability, would I be able to come into a clinic and get a personalized genome figured out for my cancer? Is that the goal?
So this field needs a lot of collaboration. We are putting together the analysis expertise around this, people who understand genomics, but at the same time, they understand high performance computing and software engineering. But we still need to work with doctors for the eventual interpretation. And what is most difficult today is the interpretation part, because the data is just becoming aggregated. And again, as I said, genomics is the main part of it, but you need to put in the context of family history, lifestyle, electronic medical records, and clinical trials. So I think we will not work with end customer or patients, but we are going to work with clinics and places for enabling that interpretation.
Sure. And then, we have a bunch of HPC users out there in our listening audience. You mentioned that there's a lot of optimization going inside your appliance. Are there any accelerators or FPGAs? I mean, how are you getting that kind of speed up? Can you share that?
Sure. So if you can show my slide. Let me see, slide number 14. That doesn't have a number, but it's after slide number 13. Talks about the architecture of the product. So again, as I was mentioning, it's a holistic approach. The innovations are happening at three layers. The first thing is the algorithms. We have our own new algorithms that are very fast and efficient. We are actually publishing a paper with Stanford University on one of our algorithms in the next few weeks. The other part of the innovation happens in system software that I think is extremely important to address the data flow problem. The way we are using the storage, the way we are using the memory, how we stream the read, how we make sure the communication is overlapped with computation is drastically improving the performance and hardware as well. So we focus, when we do all of these things, eventually we focus on the compute kernels. We see what are the algorithms or the compute kernels that most of the time is being spent on, and then we try to accelerate them. So in accelerating them, we use all the available technologies from multi-core CPUs, graphic processor units, and FPGAs. And we have currently all the three of them available in Bina Box.
And then kind of a wrap-up question here. When you sit down with potential customers, what's your toughest challenge? I mean, do they already understand the potential benefit of being able to crunch these genomes so quickly? Do you worry about financing? I mean, what's keeping you up at night?
Many things. As a startup founder, many things keep me up at night, but still I'm extremely passionate, excited about it. The challenge in terms of go-to-market for us is the inertia, is a little bit about inertia inside the market to embrace the change, because our customers, all of them, they are trying to solve this problem themselves one way or the other. And as I said, they are using open source solutions, they are trying to stitch them together and create a pipeline that is inefficient. But when we go and talk to them, using our product means to give up what they have built. And this is one challenge we are facing. The other challenge that we are facing is about how fast this field is changing in terms of the data and types of questions people want to ask from the data. And always the automation and performance comes at a price on flexibility. So there's this trade-off, how flexible you want your pipeline to be versus how fast you want it to be. And we try to find the right trade-off, but it is difficult. And we think, especially for the researchers who really like to work with open source and dig deep and like change the code themselves, it is hard to provide enough performance for them. But I think as the market moves more towards the biologists or clinicians who don't like to write code, that challenge will kind of disappear and decreases.
Well, terrific. Well, Narges, I want to thank you once again for coming on the show today.
Thank you very much for having me, and I look forward to following your podcast from now.
Okay, we'll look forward to that. Good luck with the new venture. So, okay, folks.
Thank you very much.
Yeah,
Goodbye.
you bet. Okay, folks, that's it for the Rich Report. Stay tuned for more news and information on the world of big data.